Hello, my name is Colby James, and today I'm going to teach you about replication, transcription, and translation. The goal of replication is to produce two daughter DNA molecules from a parent molecule. Replication occurs in a semi-conservative manner, meaning that one strand of the newly formed DNA comes directly from the parent DNA molecule. Replication occurs in two steps: initiation and elongation. The first step is initiation. To begin initiation, an initiator protein binds to the origin of replication, leading to unwinding. DNA helicase is responsible for the unwinding of the helix, forming what is referred to as the replication bubble. Single-strand binding proteins are responsible for keeping the DNA helix open and to protect the single-stranded DNA from forming secondary structure. Initiation of the synthesis of new DNA chain requires a short string of nucleotides called a primer. The primer is synthesized by the enzyme primase and is made of RNA nucleotides. The function of the primer is to provide a free 3' hydroxyl for DNA polymerase to work with. The second step of replication is elongation. During elongation, the correct nucleotide sequence is copied from the template strand to the newly synthesized strand of DNA. DNA polymerase III catalyzes phosphodiester bond formation between adjacent deoxyribose nucleoside triphosphates in a 5' to 3' direction. Because the newly synthesized strands are synthesized antiparallel to their respective template strand, the process of elongation occurs differently on each strand. In one strand, referred to as the leading strand, DNA synthesis is continuous. However, on the other strand, known as the lagging strand, DNA is synthesized using small fragments of nucleotides known as Okazaki fragments. DNA ligase catalyzes the formation of phosphodiester bonds between adjacent Okazaki fragments. DNA polymerase I functions to replace the RNA primer with DNA complementary to the template. The next topic we will discuss is transcription. During transcription, the enzyme RNA polymerase transcribes DNA to produce an RNA transcript. In eukaryotes, the RNA transcript is referred to as a pre-mRNA transcript, as it will be processed to become mature mRNA. The template strand of DNA is complementary to the mRNA transcript. The RNA-like strand has the same polarity and sequence as the mRNA, except that thymine is replaced by uracil. Transcription is divided into three phases: initiation, elongation, and termination. For my video, I'm going to illustrate the transcription process of prokaryotes. DNA contains sequences known as promoters that provide the signal to RNA polymerase to bind and begin transcription. During initiation, RNA polymerase binds to promoter sequences located near the beginning of a gene. The sigma factor binds to RNA polymerase to form the RNA polymerase holoenzyme. The region of DNA is unwound to form the open promoter complex, and phosphodiester bonds are formed between the first two nucleotides of the mRNA transcript. At this point, the sigma factor is released and elongation is ready to take place. Elongation is the process of constructing the RNA copy of the gene. When the sigma factor separates from RNA polymerase, we are left with only the core enzyme. The core RNA polymerase enzyme loses its affinity for the promoter and moves in the 3' to 5' direction along the template strand. The mRNA is transcribed in the 5' to 3' direction by adding nucleoside triphosphates to the 3' end of the nascent mRNA. This process continues until a termination sequence is reached. Terminators are RNA sequences that signal the release of RNA polymerase and the end of transcription. There are two different types of terminators in bacteria. There are extrinsic terminators, which are dependent on the Rho factor for termination, and intrinsic terminators that are independent of the Rho factor. Intrinsic terminators are cytosine-rich sequences that usually form hairpin loops due to complementary sequences. Translation is the process of converting the mRNA to a polypeptide chain in order to make proteins. Translation takes place on ribosomes that coordinate the movement of transfer RNAs carrying specific amino acids. Translation occurs in three phases: initiation, elongation, and termination. First, I will cover the initiation phase. mRNA contains three nucleotide sequences called codons that signal for a tRNA molecule carrying a specific amino acid. There are three sequential steps in the initiation phase of translation. First, the small ribosomal sum unit recognizes and binds to a sequence known as the Shine-Dalgarno box and the initiating codon. The initiating codon contains a sequence AUG and is complementary to the anticodon UAC that is present on the tRNA carrying the amino acid formylated methionine. The formylated methionine tRNA is positioned in what is known as the P site. Following this, the large ribosome unit binds and elongation is able to occur. During the elongation phase, the ribosome moves towards the 3' end of the mRNA, and additional tRNA molecules carrying amino acids are positioned into the A site of the ribosome. The tRNA molecules are ushered into the A site by elongation factors. The enzyme peptidyl transferase then removes the amino acid from the tRNA in the P site and attaches it to the C terminus of the amino acid located in the A site. The ribosome continues towards the 3' end of the mRNA, ushering in tRNA into the A site. As the ribosome continues to move down the mRNA, the tRNA molecules that have had their amino acid removed are ushered out from the E site. This process continues until the stop codon is reached and the termination process begins. Stop codons do not have an anticodon corresponding to a specific amino acid. The three stop codons are UAG, UAA, and UGA. When one of these sequences is reached, a release factor binds to the stop codon. The release factor releases the ribosomal sum units, mRNA, and the final polypeptide product,